The next practical in applied microbiology, course number 1, is determination of minimum inhibitory concentration of antibiotic. Whenever we say antimicrobial compound or antibiotic, it can be against bacteria or fungi or yeast, molds or yeast. In this practical, we'll learn about a common method for the determination of this minimum inhibitory concentration of that particular antibiotic for the given indicator organism. This practical is basically important in relation to clinical microbiology. You must have learned about the determination of antibiotic sensitivity test. So antibiotic sensitivity test is related to knowing which of the antibiotics which are commercially available, among which the most effective antibiotic, it can be identified by using antibiotic sensitivity test. So that practical is connected to isolation and identification of bacterial pathogen from the given urine sample or stool sample or pus sample or sputum sample. Once the organism is isolated, that organism is taken, and the next step is to determine which antibiotic is effective against that organism. So that can be done by using antibiotic sensitivity test. So antibiotic sensitivity test will tell you, or it will help you to identify the most effective antibiotic for the treatment of infection caused by that particular pathogen. Then, what is the next step? Next step is the determination of the dose of antibiotic to be given to the patient in order to treat the disease. So, for that, there is a method to decide that helps us to decide the dose of antibiotic, and that is determining minimum inhibitory concentration. So it will be the minimum concentration of antibiotic required to inhibit the growth or to kill that particular organism. So the aim of this practical is to determine minimum inhibitory concentration, that is short form is MIC. To determine MIC of streptomycin antibiotic required to inhibit gram positive bacterium. So in this practical, we'll take an example of streptomycin antibiotic and will determine what will be its minimum concentration to inhibit the growth of an indicator gram positive pathogen. Instead of streptomycin, you can consider any other type of antimicrobial compound. This will be just an example. So MIC is in vitro, that is in laboratory sensitivity test to determine the microbial sensitivity to the antibiotic. So what is the importance of or significance of this MIC? Determination of MIC will provide a guidance, or it will provide a guide to the therapeutic use of antibiotic by offering a presumptive prediction of probable in vivo response. That means we decide the therapeutic use means the dose of antibiotic, the concentration of antibiotic that can be predicted to be effective in to generate in vivo response, means to be effective against that particular infection. Sensitivity test, that is antibiotic sensitivity test, it helps in the initial selection of antibiotic. That is what I told you, as a therapeutic agent, and also help to detect any change in antibiotic sensitivity. This analysis it begins with first step, isolation and identification of the causative agent of that disease, for example bacterium. Second step, antibiotic sensitivity test, that is to select the particular antibiotic. So this first one and second one you have performed in clinical microbiology course. Third one is to determine the concentration of such antibiotic, that is MIC. And the fourth one is determination of the dose of antibiotic to be administered or to be given. It may be orally, it may be intramuscularly, or it may be intravenously. So the definition of MIC is MIC of an antibiotic is the minimum or lowest concentration of antibiotic that inhibits the growth of test organism, that is the test pathogen. Pathogen word is very important, under experimental conditions favorable for the growth of test organism. The factors that affect MIC are the time of contact of antibiotic with the indicator microbe, the temperature, nutritional environment required for the growth of pathogen, type and number of cells of indicator microorganism used in vitro testing, the nature of agent means the physicochemical properties of antibiotic and its potency, that means its power of killing or inhibiting the growth. Now, the method. In general, there are two methods by which we can determine minimum inhibitory concentration of antibiotic for the particular pathogen. First is the use of liquid growth medium. In this method, we make use of a serial dilution tube technique, which we have learned in theory in the last semester under the quantitation of fermentation product. So it is also called as tube dilution technique, which is carried out in growth liquid medium. And another method is use of solid growth medium. So in both ways we can perform the determination of MIC either in liquid medium or on solid medium. Using solid medium is a routine option nowadays. So we know that nowadays ready-made antibiotic discs of various concentrations are available in the market. So they have a fixed concentration of antibiotic, means a single antibiotic disc will have a different types of disc in which there will be different concentrations of antibiotic. Nowadays we have a single strip on which single strip of antibiotic, single antibiotic on which we'll find different concentrations of that antibiotic. So these discs are ready to use for the test. So that saves the time. Therefore, MIC it can be determined by disc diffusion method also. This is how the method is. Here we can see this is the growth medium agar plate on which you can see this green colored organism is grown. So the first step in this method is you got a growth medium agar plate, of course it is sterile. Spread your indicator organism by spread plate technique. You know how to do it with spread plate technique. After spreading, allow it to dry for few minutes, and if the culture is dried after 10 minutes, say for example, then you can see this is the antibiotic strip, and you can see the lines over here and some figures are marked, so it is not clear, but you can see that here it is high concentration of antibiotic. Then it will go in, if you go in this direction, the concentration of antibiotic decreases. So this is a single antibiotic strip with different concentrations of that antibiotic. Now the next step is to place this strip onto the plate, and after that this plate A will go for incubation at 37 degrees Celsius for 24 hours. After 24 hours of incubation, the picture will be like this. You will find the zone of inhibition. So here this antibiotic will diffuse in the medium, and wherever antibiotic is present in high concentration or the sufficient concentration to inhibit the growth, you will find that the growth is inhibited. So this is the high concentration where the growth is inhibited. If you go down, you are going down. As you are going down, the concentration is decreasing, and you can see this point which is shown by this arrow, that this is the concentration. That means this is the highest dilution of antibiotic, or this is the minimum concentration of antibiotic which shows inhibition. So this is minimum inhibitory concentration of that antibiotic. That means it is the lowest concentration of that antibiotic which still inhibits the growth of indicator organism. So this value is your MIC, the concentration of antibiotic. This is how the solid medium is useful for the determination of minimum inhibitory concentration. Very easy technique and very reliable and time saving technique. Now we'll discuss about tube dilution method, in which we'll see exactly how this tube dilution method is useful to determine MIC of streptomycin. So streptomycin injection, which is available in the market. Now this is the vial which contains 0.75 grams of streptomycin. You can convert this into milligrams, and further you can convert that into micrograms. The requirements for this tube dilution method are streptomycin vial, that is ambidextrine, sterile phosphate buffer for the dilution, sterile nutrient broth tubes for the growth of indicator bacterial pathogen, 5 ml in each tube. Then 24 hours old culture of the test organism. Here will be determining minimum inhibitory concentration of streptomycin for Staphylococcus aureus, the gram-positive bacterium. We'll need sterile pipettes and sterile saline. Now, the procedure. First step is to prepare the stock of streptomycin antibiotic, that is working stock. We need that working stock in this concentration 100 micrograms per ml. We already learned how to prepare stock of antibiotic in previous practicals. So it will be just a revision for you. This is the antibiotic vial that contains 0.75 grams. That means it contains 7,50,000 micrograms of streptomycin. So drop this 0.75 grams, that is 7,50,000 micrograms, and mix it with 100 ml of sterile phosphate buffer. So 7,50,000 micrograms in 100 ml. What will be its quantity in 1 ml? That will be, so 7,50,000 micrograms corresponds to 100 ml. 1 ml corresponds to how much? That will be 7,500 micrograms per ml. We already learned this. This is called as stock A. Then we'll go for preparation of stock B. From this we'll prepare stock B, that is 100 micrograms per ml. Now we want to prepare this in a quantity of 100 ml. So we have the formula RT by G. R is required. Required is this. T is total. Total is we want to prepare 100 ml of this concentration. And G is this given 7,500. So if you do that for calculation by using that formula, the value will be 1.33 ml of stock A you had to take and make volume to 100 ml. That will give you a stock B having concentration of 100 micrograms per ml. Now this stock B having 100 micrograms per ml, it will be your working stock. Now this is the stock B. You have sterile tubes. All these tubes are sterile. Mark these tubes as 1 to 10. So this indicates the concentration of streptomycin. These many dilutions you have to prepare. From 1 micrograms per ml to 10 micrograms per ml. Now the question is, why from 1 to 10? Answer is, it can be anything. You can take 10 to 20, high concentration. You can take 0.1 to 1, 0.1, 0.2, 0.3, 0.4, 0.5, 0.6, 1. So you can take in this zone, that is a low concentration zone, or you can test in high concentration zone. Through the practice we have determined and experience we have determined that MIC of streptomycin will come in this range, and that is why we have taken this 1 to 10. In practical, if you don't know anything about MIC, you should start from the low concentration zone with broad range. Low concentration with broad range means you can take 0.1 micrograms per ml, directly 0.5 micrograms per ml, 1 microgram per ml, 2 microgram per ml, 10 microgram per ml, so on and so forth, so that you can take a broad range. And once you determine that MIC is less than 1, then you can further test it from 0.1, 0.2, 0.3, 0.4, 0.5, 0.6 and 1. If your MIC is coming less than 10, it is coming between 5 and 10, then you can think about the narrow range or within 5 to 10. We can take 5 5.5, 6, 6.5, 7, 7.5 up to 10 likewise. So this is how we can decide the range. Now we'll be testing MIC between 1 to 10, say for example. So what are these tubes containing? All these tubes are containing liquid growth medium, that is nutrient broth. How much nutrient broth is present here? 9 ml of nutrient broth. Okay, remember that it contains 9 ml of nutrient broth. And after the addition of antibiotic solution into this, the total volume is going to be 10 ml. That means we'll be adding 1 ml of antibiotic solution in all these tubes. So the total quantity is going to be 10 ml. Now, like we have prepared a dilution scheme in case of antibiotic assay, here also this is something same to that protocol. Here also we are preparing a dilution scheme. What is the dilution scheme? The first tube having a concentration of 1 micrograms per ml. So how will you calculate that? Simply by using a formula of RT by G. What is R? Is 1. T is this one, 10 ml. And what is G is given, that is 100. So 1 into 10 divided by 100, the answer is 0.1 ml. 10 by 100 is 0.1 ml. Okay, this means that you had to take 0.1 ml of stock B and add it to first tube. So what will be the total volume? It will be 9.1. We have to make the volume to 10 ml because here we are consider this value of T as 10. So what will be the remaining amount of liquid? It will be added in the form of sterile phosphate buffer. How much? 0.9 ml. So got this composition of this tube: 9 ml of sterile nutrient broth, 0.1 ml of stock B and 0.9 ml of sterile phosphate buffer. This is how you can calculate it for tube number 2. You can do this on your own. Simply pause this video and you can calculate on your notebook. So if you calculate it for this tube number, second tube having concentration of 2, we want concentration 2, that is R is 2, total is 10, given is 100 and the answer will be 0.2. This means that in this tube you have to add 0.2 ml of stock B plus 0.8 ml of buffer. Likewise, you can calculate for this 3 micrograms per ml tube, 4, 5, 6, 7, 8, 9 and finally for 10 micrograms per ml it will be 10 into 10 divided by 100 and the answer will be 1 ml. So in this you have to add 1 ml of stock B and no phosphate buffer. This is how you have prepared these dilutions. Now we'll be having 2 more tubes along with this. These tubes will be called as controls. What are these controls? We want to compare the result of this with control. One will be positive control, another will be negative control. Now see, this is one control in which there is 9 ml of sterile nutrient broth is there plus 1 ml of sterile phosphate buffer. This will be called as clarity control. So only nutrient broth is there. Along with that, there is presence of buffer. Sterility control or clarity control. Second tube will contain 9 ml of sterile nutrient broth plus 0.1 ml of suspension. That means the suspension of bacterial culture. It will be called as turbidity control. Now you have these two controls. One is clarity control and the another one is turbidity control. This means that you have to compare. Turbidity control will tell you how exactly the growth of this test organism appears, and clarity control will tell you if there is absence of growth, how that particular tube should appear. This is the dilution scheme that you need to prepare before going for this step. So first we have to prepare this dilution scheme. Write down tube number, first column, second column, concentration of streptomycin in micrograms, stock solution in ml, sterile buffer in ml, sterile nutrient broth in ml, total volume and bacterial suspension. Now number these tubes like 1 to 10, 11 and 12. Now concentration, as we have seen, 1 to 10. Now this part we have already calculated. The concentration, the quantity of stock B, and how much buffer has to be added. This is common in all the tubes. There will be 9 ml of sterile nutrient broth. This is common for all. In all the tubes there will be the total volume of 10, and in all the test tubes, that is from 1 to 10, you have to add equal number of cells, the test organism, that is 0.1 ml of suspension. This is a clarity control, so there is no need to add suspension. The last one is turbidity control, so we need to add suspension. All these tubes after addition are then incubated. After incubation, the picture will be like this. You have to observe the controls first, then it will tell you whether these results are correct or not, authentic or not. Turbidity control should show the presence of growth. Clarity control, there should be absence of growth. Then you can say the rest of the tubes are showing authentic results. You can see in this that this is low concentration, this is high concentration. This is low concentration means higher dilution of antibiotic. Here high concentration of antibiotic, lower dilution of antibiotic. Okay, so obviously when we go from low concentration to high concentration, you will find that the growth of the test organism will decrease. So you can see in this result, tube number 1 is showing turbidity. Tube number 2 is showing turbidity, means dilution of antibiotic is not sufficient enough to inhibit the growth. This concentration is not sufficient. This concentration is not sufficient. This concentration is not sufficient to inhibit the growth, but the turbidity is decreasing. This concentration is not sufficient, but at this 8 micrograms per ml, you can see absence of growth. There is absence of growth. That means this is the concentration which is sufficient to inhibit the growth of test organism. So what is the result? Is it that the result is MIC is 8? No. If you see this 7 which is showing turbidity and 8, so there is a huge big range between this 7 and 8. What is that? 7.1, 7.2, 7.3, 7.4, 7.9, 8. So have we tested that? No. Therefore, we need to report this MIC that MIC lies between 7 and 8 micrograms per ml. So the MIC of streptomycin is found to be between 7 and 8 micrograms per ml. That is your result. Or you can say MIC is more than 7 and less than 8.